Some of the most important developments recently is the ability to analyze thousands of cells. So single-cell genomics started by analyzing, let's say, tens and hundreds of cells, and very quickly it has moved towards thousands and 10,000s and now 100,000s of cells. So I think one of the most important development has been droplet-based technologies. Droplet-based technologies that allowed these numbers of cells to be analyzed. And now we are moving towards combinatorial approaches that allow 100,000 to possibly even 1 million cells to be analyzed in parallel. I can tell from my lab's experience, so from our perspective, they are twofold. One, by analyzing tumors, identifying the heterogeneity of the tumors and subpopulations, and then using the transcriptomic or genomic information to identify targets that can be used to guide the therapeutic approaches. So that one thing would be analyzing the tumors themselves. Another important application in our case is using patient-derived xenografts that are then analyzed, and performing single-cell genomics on xenografts allows us again to identify possible targets that are population-specific, and the aim of everybody is actually to target the stem cell populations, which are the ones that drive tumor growth. So these are the two main applications that we're working on at the moment for developing therapeutics. The tools can be divided into three types. There are instruments that allow basically us to capture single cells, and there have been a lot of developments in this area. A widely used tool, for example, launched by 10x Genomics, allows high throughput capture of cells at the 1,000 to 10,000 level. So this instrument has proven itself to be quite reliable and it's been widely used. But there are competitor instruments that are worth considering as well. The other important development is the bioinformatic tools to analyze the data. Capturing the cells and generating data is one thing. Bioinformatic analysis is another thing. And recent developments like using tools like RNA velocity, for example, has allowed us to gain completely new insights into how cells develop from one stage to the next. That's one thing. And the other thing that we should not ignore is the development of suitable molecular biology that allows one to perform either 3' type RNA expression profiling or full-length cDNA synthesis and sequencing, but also to analyze the genome of the cells through whole genome sequencing approaches. I think that there are a couple of things that are very important and are all in development. One would be to increase basically the number of cells that are captured at a very low cost, and I think combinatorial approaches are leading the way at this point. The other important aspect, in my view, is how we bring spatial information to single-cell genomics, and approaches like StarMap that has been published recently that allow us to analyze the transcriptome, for example, of cells or even proteins, and retain the spatial information are going to be critical in understanding tumor heterogeneity, but also differentiation processes in all types of life forms.